louder.
The family medical history.
Okay, so maybe, yeah, maybe if they do family medical history on for all of the families, yeah. There might be some interacting factors. Okay. So for this, so it turns out that back in the 80s, they did not actually require, I know now when we purchase Tylenol or any medication over the counter, it has a seal, right? You probably are familiar with that all medication is sealed. At the time in the 80s, that was not required. So you could buy over-the-counter medication and just open it up in the store. And so after this event, they actually changed legislation to require that, so that now all medication has to. So FDA changed that requirement after this, because in fact somebody had tampered with the Tylenol there that all of these individuals took. So for part 2, when they looked at the autopsy and they're still trying to track down what could have been the cause, like what exactly was added to the medication, they found that the patients had hypoxia, evidence of hypoxia. So when you look up hypoxia, what does it say? What is the definition of hypoxia? Go ahead, Matt.
Deficiency of oxygen going to the tissues.
Okay, yeah, so deficient oxygen levels in the tissues. And then it said that their blood, oxygen in their blood was 110 millimeters mercury. What did you find as being the normal oxygen level for blood?
75 to 100 in a machine.
Okay, so this is odd. Why did they have high oxygen but hypoxic tissues? Anybody have any ideas?
Aerobic cell respiration was interrupted.
Yeah, so part of what we need oxygen for is for cell respiration to be the final acceptor. So somehow that was not happening. Whatever this poison was, it was interfering with cell respiration. So there is oxygen, it's just not making its way into the cells. Okay, so then if you flip it over here, when they did toxicology, they found that all the victims had cyanide poisoning. So there was evidence of cyanide poisoning. Somebody, I don't remember why this person, like what the motivation was for doing this, because it seems like extremely random to poison random people. But somebody had tampered with the Tylenol and added cyanide. Cyanide is very quick acting, and it can kill, depending on dosage, can kill within minutes of ingestion. I was reading on the poison control site about cyanide poisoning. Cyanide can be found in other places, like very small amounts, trace amounts. And sometimes people get exposed to it over a length of time if they work with certain substances, but for the most part, most of us should not encounter cyanide, right? So the way that cyanide works, if you look at this picture here, it binds to the 4th enzyme, or the 4th complex, which is this complex right here. The 4th complex in the electron transport chain. And that 4th complex is actually the complex that hands off the electrons to oxygen. So if you kind of think of the electron transport chain like what did you say? Like an assembly line or something. This is the final step of the assembly line, and it's basically the place where you hand off to oxygen. So it makes sense that if you stopped, or you kind of put a kink in this chain reaction at that step, that the electron transport chain is going to entirely stop altogether. That you would not have movement of the electrons through the electron transport chain. If you don't do that, you're not going to generate the proton gradient. So we're going to talk about how this works, and then we'll come back to this picture, and hopefully you'll be able to label all the parts of that by the end of our discussion on electron transport chain. So we'll do number 4 in a second, in a few minutes. But for number 5, now can you explain why there was increased oxygen in the blood? Yeah.
Because oxygen wasn't binding to the carbon molecules to take them out.
Yeah, it wasn't accepting any electrons, so because you had stopped this entire flow of electrons through the electron transport chain, it was building up in the blood, and it wasn't being taken into the tissues. And then for the last one, this is just kind of an interesting fact about cyanide. So back in World War II, they used cyanide as a suicide pill. So if you had spies, British and American spies, if they wanted to basically commit suicide so they wouldn't have to tell or give any information up, they carried with them this L pill that they would take, and it was a cyanide pill, and it would cause them to die immediately. And so the question here is that, you know, based on what you know about the need for ATP and how much ATP we have on reserve in our body, why would you think cyanide would be so fast acting? Yeah, go ahead.
Because we only have a little bit of ATP, like just hanging out in reserve at one time, and so we need aerobic respiration in order for it to create more.
Yeah, exactly. And if you think about different cell types, what cell types do you think have the highest ATP demands in the body? Muscles are, what's a very important muscle that we have? Our heart, right? Neurons and muscles, heart, are two of the highest demands. And so obviously if we kind of deplete all ATP and are not creating more than it is gonna cause a pretty quick death if the dosage is high enough. Interestingly, if you get cyanide poisoning, they do have antidotes for it, and one of the ones I looked up on poison control, I'll tell you the exact name of it. Hopefully you'll never need to know this, but they use hydroxycobalamin, which will actually compete with complex 4 to actually bind cyanide. So it will sequester the cyanide or bind it up so that the cyanide can't bind to your electron transport chain proteins. So if you ever suspect cyanide poisoning, you have any of these symptoms, then anyway, that's what you can do for treating a cyanide exposure. Okay, so we're gonna fill out this box number 4. Let me now switch over back to our PowerPoint for the electron transport chain.
I already did that.
What did you say?
Box number 4 yesterday.
Oh, for number 4 here. Well, if you have that information, you're welcome to fill in the labels on this one. But yeah, if you haven't yet, we haven't yet talked about electron transport chains, so. Okay. So we left off, we went through, we talked about what we have so far yesterday with this chart. We already added up how many NADH we have and how many FADH2 have been produced. Anybody, if you want to look back at your notes, what was the number we got for number of NADH that have so far been produced? 10. So we've got 10 NADH, we have how many FADH2? 2 so far. And how much ATP have we net created so far? 4 total, right? We did 2 in glycolysis and 2 in the citric acid cycle. So that's what so far we've made. Now, if we know that the total ATP that we're going to create theoretically on the second question here says, here's the theoretical yield based on how much proton motive force can be created by the NADH. What number did you get for the total ATP theoretically that could be produced by 10 NADH and 2 FADH2? 34, just from the ETC. So the last stage, we should be able to make 34 ATP, plus we already have 4 from glycolysis. So when you see the number in a lot of textbooks, they will say 38. Sometimes they will estimate 36 to 38. They take off 2 because you do have to transport NADH into the mitochondrial matrix. So that's why you see this range. In actuality, we're not going to create that much because the proton motive force is not entirely used just for the purpose of making ATP. It is used for other things as well. That's one reason. Second reason is that biological systems are not usually 100% efficient. So we won't create in actuality that much, but we'll get pretty close. So this is the overall summary for electron transport chain. Our goal is that we still have these electron carriers that we can harvest energy from. So the goal will be to use the energy from those electron carriers, and we're going to make a gradient that can then be used indirectly to make ATP using ATP synthase. So this is where the oxidative phosphorylation comes into play. The location for this, we kind of oriented ourselves with what the mitochondrion looks like. You have outer membrane, inner membrane, matrix, and the IMS, right? All of this is taking place along the inner membrane. And part of the reason that the inner membrane is so curvy is to increase surface area. So you have a lot of surface area for these electron transport chain proteins to make ATP. Okay, so the inputs are going to be all of the electron carriers that we've made so far. 10 molecules of NADH, 2 of FADH2, and we have to have oxygen present because oxygen is our final electron acceptor. The output, the numbers I put here are for actual and not theoretical. So we calculated we should be able to get 34, but actually we get something closer to 24 or 25 molecules of ATP. But even still, that's most of the ATP made, right? So far we've only made 4, so that's still the majority of the ATP that is created. And then we also produce water. So the water, can anybody actually see where the water would come from? How the water is made? When oxygen takes electrons, what else is going to go with the oxygen, electrons? Protons, yeah, hydrogen, and that's going to create the water. Okay, so we've already gone over this and that. So let's talk now. So we're kind of zooming in. This picture right here, let's see if they put it in it. Yeah, they drew it in this orientation. So if we were to take this section and zoom in on it, it would look like this. So here's the inner membrane space, here's the mitochondrial matrix. Along that, we have transmembrane proteins. There's 4 different complexes that are embedded. You can see them here. Complex 1, 2, 3, and 4 go across the membrane. So those are transmembrane proteins. There's also mobile factors, factors that can move. The orange one right here, it says Q. Q stands for quinone, ubiquinone. Okay, that's one name for it. You also will see it called coenzyme Q. And sometimes they sell, or CoQ10. CoQ10. You'll, at Costco actually recently, I was walking through and they were giving supplements for CoQ10. People take it as supplements to reverse the effects of aging. It's supposed to help your heart and all these other things. So, ubiquinone is, I want you to make a note of this. This is actually a lipid. It is not a protein. It's shown as sort of a purple circle or a orange circle here, but it is actually a lipid. And what do we know about lipids? Are they hydrophobic or hydrophilic? Hydrophobic. So it does make sense that this ubiquinone, as you can see it here, is able to move across the membrane. No problem, because it's a lipid. So it'll be involved in carrying electrons from one side to the other. And then the other mobile factor, you see, is right here, cytochrome C. Cytochrome C, it does not look like it's going through the membrane. No, so it's a peripheral membrane protein. It's not transmembrane protein. It goes in between complex 3 and complex 4. It's like a little ferry. It's going to pick up electrons and carry them from 3 to 4. And it goes back and forth, carries 1 electron at a time. Okay, so that's sort of the basics of the electron transport chain. You should be able to identify. I will not ask you to draw them, but if I drew them, or I had an image with them, you should be able to label them. So you should be able to say this is complex 1, 2, 3, and 4, and then also label ubiquinone and cytochrome C. All right, so the goal here is going to be to pick up electrons and pass them along. Like I said, it's sort of like an assembly line. They're going to go from complex to complex. And I'm going to tell you the path that they go for each. For the electrons that get dropped off by NADH, they have a different path than the electrons that get dropped off by FADH2. You can kind of see that right here. First of all, where they're getting dropped off. So we're going to start with NADH first. As NADH hands off its electrons, you can see it becomes NAD+, right? So what has happened is NADH is now oxidized. It gave up its electrons, and its electrons are handed off to complex 1. So then what would we say complex 1 is oxidized or reduced? If it takes those electrons. It's reduced, exactly, and then it's gonna, you can see the electrons go to ubiquinone as soon as complex 1 hands them off. Then complex 1 is oxidized and ubiquinone is reduced. So it's a series of oxidation reduction reactions where the electrons just move from complex to complex. The other thing you notice about this is that at certain steps, you see protons shooting out into the intermembrane space. What would be the purpose for this? Why do we want to send all these protons into the intermembrane space? Yeah, we ultimately want to have an electrochemical gradient so ATP synthase can use that gradient to make ATP. So this is how the electron flow is indirectly going to be tied to producing ATP. All right, so we're going to start at the beginning. So the beginning is those electrons that came from NADH, their path. They get handed off. We'll write the path up for NADH. Electron flow path goes from NADH, they get handed to complex 1. From complex 1, you can see they go to ubiquinone. Okay, so NADH, so this is kind of the steps. NADH is oxidized as it hands its electrons. The electrons go from complex 1 to ubiquinone. Just put Q. And at that step, you pump protons. I don't care if you know numbers of protons, and we're not going to kind of account for that. Okay. Do this later. All right, so we have protons are being pumped in complex 1 as the first step. Now, does it look like that Q goes to complex 2 in the picture? No. So it skips and goes to complex 3. So the electrons that come from NADH, they never stop at complex 2. They go from complex 1 to Q to complex 3. Okay? Oh, no, now they, Adobe update. Okay. Oh, and I put that, oh, I have this on the side too. The CoQ10 supplements, and they also put them in creams because this is a very powerful antioxidant. Which, so some, people want to reverse the effects of aging by taking supplements of an antioxidant. So that's kind of the thought behind it. It's also supposed to, like I was saying, the CoQ supplement is thought to reduce, what does it say, reduce the likelihood of a heart attack, a repeat heart attack. So if you know people who have already had a heart attack, they potentially might take CoQ10 supplements. Okay, but now on to, I'm going to skip this slide because we'll stick with our path with NADH and then come back to the path with ADH2. So complex 3 here is accepting electrons from ubiquinone. Now one thing you notice about ubiquinone, in addition to passing electrons, it's also picking up a proton. So it does it picks up a proton with the electrons and it carries them and drops them off on the intermembrane space. So it's also contributing to the electrochemical gradient. It passes the electrons from complex 3, so from Q to complex 3, and again, complex 3 pumps protons, 4 protons. And then, cytochrome C has to go back and forth between these two. So as you can see, because these are the transmembrane proteins can't move, they need something to move in between the complexes. So cytochrome C picks up an electron one at a time and carries them from complex 3 to complex 4. And again, complex 4 we pump 2. So total there was 10 protons pumped through all of these complexes. Okay, so complex 3 goes cytochrome C and then to complex 4. The last place where the electrons go is oxygen, final acceptor. And so make sure you know this because when we talk about photosynthesis, it'll be different what the final acceptor of the electrons is. But the final acceptor will be oxygen. And as you can see, what happens when oxygen accepts the electrons and the protons, it becomes water. So that creates the water byproduct that we see with cell respiration. Okay, so any questions so far on the pathway? So you should be able to label these and also show what is the pathway for the electrons. All right, so going back to FADH2. FADH2 cannot hand off at complex 1. Why would that not be able to hand off? It has to do with the free energy levels. It has to do with the free energy change. Okay, so you see that NADH is way up there at 52 kilocalories per mole. FADH2 is lower. Because it's lower, it's not above the level, the free energy level of complex 1. That's why you can't hand back up to a higher free energy level. So that's why FADH2 hands off its electrons to complex 2 instead. So for this, come back to this slide in a second. Now we're going to do the path for FADH2. So these FADH2 electrons, they go to complex 2 instead of complex 1. They'll never go to complex 1. But complex 2, you can see, can also hand off to Q. So at this point, it hands its electrons into the same pathway. And everything from that point forward will be the same for those electrons. Okay, any questions on just the general path for the electron transport chain? Okay, so as we are going, and this shows you in detail which component of the complex is taking the electrons. You don't need to know that. I just, big picture want you to know the complexes, not the actual, iron sulfur clusters and things like that. But as you're moving down, you can see that we're going down this free energy. The free energy is decreasing, right? So energy is being given off. So what do we do with the energy that is released as electrons are moving? What is it used to do? It's going to be used to do something that requires energy. Yeah, but what are we going to do to make the ATP? Ultimately, we're pumping protons, right? And when we're pumping them to a region that already has high concentration. In this intermembrane space, protons are being pumped into this IMS. They're already at a high concentration, and we're forcing it to get higher and higher because we want to have an electrochemical gradient. So the energy of these electrons moving through the complex, as that moves down its free energy, it's using the released energy to pump protons. So that's an example of coupling. Remember in the energy and enzymes lecture, I talked about coupling of reactions. This is coupling exergonic reaction as electrons are flowing with something that is endergonic, which is pumping protons against their gradient. So for that blank on that slide, it should be the energy released from redox reactions in the ETC is used to pump protons against their gradient into the IMS. Okay, so why do we ultimately want to do that? We said that that's what we're going to use for making ATP. So the engine that does the ATP synthesis is called ATP synthase. This is a little GIF that shows the activity of that enzyme. It's basically nature's molecular motor. So the closest thing is you can have to an engine. And I actually have a little video that explains how it makes ATP. In this picture, just to orient you, they flipped it the other way. So this is showing it with the intermembrane space down here. This thing is in the matrix side, so protons are flowing this way. They're going from the IMS down their gradient through the ATP synthase. So for the blanks for this slide, hydrogen flowing down its electrochemical gradient from the IMS to the matrix. It's called chemiosmosis. So the word, we already talked about osmosis. What was osmosis? What does that mean? What is moving in osmosis? Water. So water is moving. Chemi means that something else is moving. It's protons that are moving down the gradient in chemiosmosis. So let me play this video that shows kind of how this machine works. Oh. They always pick British guys to be the, okay, how do I end this? Oh. Okay. Just turn it.
It's been called one of the wonders of the molecular world, an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel that every one of your cells needs to function. Without this fuel, your cells will cease operation, and so will you. ATP synthase works like a rotary engine. The barrel-shaped rotator is composed of 10 to 15 protein parts called subunits. The rotator spins around, transmitting mechanical energy into the drive shaft of the machine, which helps make ATP. This drive shaft has a specially placed bump that opens and closes parts as the drive shaft spins around. This bump opens special protein subunits on the bottom of the machine. When the bottom subunits open, a spent energy molecule called adenosine diphosphate, or ADP, enters the machine. The mechanical motion causes the ADP to bind with an additional phosphate group, creating the ATP energy molecule. And the ATP drifts off into the cell, ready to power some biomechanical reaction. The ATP synthase machine has many parts we recognize from human-designed technology. A rotor, a stator, a drive shaft, and other basic components of a rotary engine. The ATP synthase is one of thousands of elegantly designed molecular machines inside your cells that make your life and all known life possible. ATP synthase. An example of intelligent design.
Very dramatic. Okay, so you kind of see what happens is that there's a bump, and as it spins, it opens up the leaflets and lets in ADP and phosphate, and then those two go together. So that's sort of how your ATP synthase works. The question I had on that slide with, where's the other PowerPoint? Oh, here it is. So here's a question: what would happen if you dissipate the proton gradient? So what if you had, and there actually have been drugs, there was a diet drug that people took in the early 1900s that is called DNP, dinitrophenol, that basically dissipated the gradient. And people lost a whole bunch of weight, but they also died, a lot of them, because they got hyperthermia. So they were giving off, they had very high temperatures. So what happens if you get rid of that proton gradient?
You don't have enough energy to make ATP.
Yeah, so you're not making ATP. Can anybody think of why they would be hyperthermic for those individuals?
Energy things.
Yeah, so instead of going through ATP synthase, it's just going down its gradient, and that energy is giving off heat. Essentially what it does is it uncouples the intake of food from the production of ATPs. That's why people lost weight on it, but then it was obviously outlawed because it was lethal in some cases where people had dinitrogenol. So, if you dissipate the gradient, that will prevent you from producing ATP. It uncouples the gradient from ATP synthase, and then you will release heat instead of using that gradient to produce ATP. Sort of the explanation of that. Okay, so it looks like we probably need a little break. So let's take like a 10-minute break, and then when we come back, we'll finish out cell respiration. We can start on photosynthesis, so most of photosynthesis is done by tomorrow, because I want to make sure we have time to review for tomorrow as well. Let's take a 10-minute break. We'll come back at 12:55.
Are we